Hello, everyone, and welcome to Synthace's Lab Automation Masterclass, running powerful automated. I'm Markus Gershater. I'm the co-founder and CSO at Synthace, and I've got the pleasure of presenting today. I thought we could all start with a bit of basic introduction and some sort of common ground. Whenever we're thinking about running an experiment in biology, then the onus is on us to think about how we're going to run that experiment. How can we best address the complexity inherent in biology and run an experiment that's going to help us to cut through that complexity and really understand what's going on behind the scenes? Now, the way that most people have done biology for most of their careers, and certainly the way that I ran my science in the earlier parts of my career and the way I was trained to do my science, is essentially one factor at a time. So this is where you choose a factor that might affect a particular process. You choose the levels over which you're going to then try and investigate that factor. You choose some points at which you're going to assess that factor at, and then run some experiments. And then you get your results from those points, from those experiments, and you draw a line through those points. And so in this case, we've investigated temperature, and we say, okay, 15 degrees must be our best temperature. And then we might go on and investigate another factor, and 25 degrees is our best temperature. And so then we'll investigate another factor, and then we'll investigate that at 25 degrees because that's our best temperature, and we'll get more data, and we'll say, okay, this is our optimum. But this is the fundamental issue, because in biology, it is a highly complex system, and it's rare that factors behave in such a predictable manner. Because in this case, what we've done is we've actually taken two cross sections through a two-dimensional space when we're running these experiments. And we won't necessarily have even thought of the fact that's what we've done, but this is what we've done in actual fact. And so what we've neglected is the fact that there could be an interaction between those two factors, and our optimum could actually lie somewhere over to the right of this graph. So instead of this, what DOE does is it sets up a matrix of different experimental conditions within high dimensional space. And what that allows us to do is to see how different factors affect the system as other factors change at the same time. The reason that we can do this is because we have a very structured experimental design, which means that we can still then deconvolute the effect of each factor, even though we're changing many factors simultaneously. So a classic kind of design is you'll see a lot of these kind of cubes whenever anybody's talking about DOE. And essentially, if we're looking at then temperature, time, and pH in this fictitious experimental system, then we have design points all the way from where all of those are set to low to where all of those are set to high, and then every combination in between. When time is low, temperature is high, and pH is low, for example, would be one of these data points on the cube. When we're designing a particular design of experiments, we choose levels that we want to investigate for each one of these. And then if we want to see what the effect of, say, temperature is on the system, then we compare the left-hand side of the cube to the right-hand side of the cube. So although when we're talking about like multifactorial experimentation and using such grand terms, it can seem quite daunting and fairly obscure, actually what we're doing is something pretty simple, which is taking averages of different readouts and comparing those averages to see what happens as a particular factor changes. So in this case, we'd average all of the results from 25 degrees and compare them with the average of the results from 37 degrees and do a simple linear interpolation and see what the effect of temperature is on this system. But what it also then allows us to do is to look at the effect of the temperature when we leave it, say, overnight or 3 hours. So then we can start to see whether we have a temperature time interaction, for example, which is actually a very common interaction that we'd see in a lot of experimental systems. In this way, we can use design of experiments to very rapidly look through a lot of different factors that might affect our system, but we can also start to see how those factors are interconnected and look at those factors and how they interplay with each other. So that's all well and good for 3 factors. So this is a 3-factor design. But biology is a lot more complex than this. So what about if we want to investigate more factors? This is my attempt at drawing a six-dimensional shape on a 2-dimensional space. So forgive me if it's a little bit complex. But you don't really have to understand exactly what this diagram is trying to say. Essentially, but briefly, what I've got is 3 more factors, which are denoted by the cubes on each of the corners of the big cube. So factors A, B, and C. But the main point that this slide is meant to illustrate is that as we add more factors, then the number of experimental runs, which are these dots, goes up exponentially. Whereas with a simple cube with 3 factors, then we can cover those points with 8 different design points. As we go up, to higher numbers of factors and higher numbers of levels, then the number of design points that we have to address goes up exponentially. So now with 6 factors, that would be 64 design points, and it goes up such that the number of runs that you have to do for any experiment is 2 to the n, where n is the number of factors that you want to address. Obviously, this becomes untenable very quickly, even if we're using automation. If you're looking at something quite ambitious, say 20 factors simultaneously, then that would be over 1 million design points, which just wouldn't be something that would be particularly easy to carry out, even with automation. In fact, it'd be prohibitively difficult. But the good news is that actually we don't have to run all of these experimental design points if we want to still gain a good understanding as to the basics of what's happening in the system. So what DOE allows us to do is to systematically strip away a number of these design points that we would have had in a complete design into what's known as a fractional factorial in this instance, which still then allows us to see the main effects that are happening within our system, and also some of the ways that those factors are interacting with each other. So that's the fundamental of DOE. And long story short, we've got a lot of previous material about this as a company. And if this is your first time hearing about DOE, then I'd encourage you to go back and look at those. This isn't where I'm going to go much further into this. Suffice to say that we've had a lot of success with this in the company. This is still one of my favorite case studies, where we can see that as we go through iterations of design of experiments in the same system, where we're exploring design space by exploring a particular region and then realizing that if we move the cube, then we should get a better response and so on, and walking our way through a system, then you can see from the results that we go from week 1 in the blue here, where we did manage to achieve a bit of expression, all the way to week 4, where that expression is unrecognizably high. So very rapid optimization of a complex biological system. This was a 15-factor experiment that we ran over 4 weeks, where there's about 64 runs per experiment. So it shows you the power of DOE to be able to cut through biological complexity. And this was all done by hand. This was early days in Synthace, before we were a software company, when we were looking to use DOE to improve the performance of various biological systems. And we got some really nice results. It then poses the question, why bother automating DOEs? Now, the answer is because there's never really an end to biological complexity. And the journey we went on as a company is that we were working with various partners, and the challenges that they'd throw at us in terms of the kind of optimizations and systems that they wanted to establish only ever got more complex. And so then our drive was to run ever more powerful experiments to meet that complexity. So when we're automating, we're thinking about classic things that you get with automation. We can have obviously higher throughput, higher accuracy, higher reproducibility, all of these things incredibly important so that we can get a decent amount of very high quality data that we can then model. But there's also something additional with design of experiments, and when we're thinking about automating design of experiments, and that's the issue of complexity. So any experiment where you're running a large number of runs, where every single one of those runs is varying over however many factors you've chosen. And we've internally run anything up to 27 factors simultaneously, and our clients have run up into the 20s as well. And so you can imagine, say, running a 300 run design where every single one of those runs varies over 20 different factors. That's got an absolutely massive level of complexity to deal with, which frankly is just very difficult for a human being to design, plan, and then execute on in the lab. So this is what one of the fundamental things that we're trying to address when we're looking at automated experiments. So let's have a think about the hardware that we can use when we're looking to automate. Let's start from that end. So a lot of the classic hardware that we'd use when thinking about automating something in the lab would be liquid handlers. So we've got a few examples here. We've got a TCAM Fluent, we've got Gilson Pipette Max, Hamilton Star, Beckman, and a SciBio as well. And all of these are tip-based systems. So they will be using those tips to pick up from source plates and then deposit those samples back into destination plates and do mixing and so on. And so they're basically mimicking what we'd do with a handheld pipette, but they can do it without any worries about how complex or difficult the protocol might be. And so there's obviously a real advantage there when we're trying to run a highly complex design of experiments. The disadvantage that we found when we're running DOEs with this kind of equipment is that especially when you're looking at low volumes, then to get accurate transfer of liquids, then you need to aspirate from your source volume, but then you need to go and actually touch the tip into the liquid that you're then dispensing into so that you actually get an accurate transfer and you don't get like the droplet hanging on the end of the tip or whatever. So then that tip's now effectively contaminated with the destination contents now on the outside of that tip, so you have to go and inject it. And again, if we think about a complex experiment where we might have 300 runs and 20 factors, then that will be 300 times 20, 6,000 different liquid handling transfers. And if you're changing your tip every single time, that's 6,000 tips. And it's also a huge amount of time. You either need to think about ways that you can work around that, or you're limited to a maybe less complex type of design. On the other hand, you can also think about a different type of liquid handling automation, which are the dispensers. And we found with Synthace, at Synthace, as we've run many DOEs in our own lab and seen what's possible at clients as well, that these dispensers offer a disproportionate power because they're dispensing from above, or in the case of the Echo, which is the machine down in the bottom right of the screen, it's dispensing up into a plate. But either way, there's no contact between the heads or the tips or whatever and the destination wells. So what that means is there's no need to eject tips if there are tips on the system. And so it means that you can just have a very rapid dispensing of a large number of different liquids into your destination plate without having to worry about consumables usage to the same degree at all, and also at a much faster pace. As with any automation system, each of these has their own strengths and weaknesses, which can be used depending on the type of DOE that you're running, but also on the kind of biology that you're doing and what volume you can run that biology at. These aren't really kind of direct like-for-like competitors. Really, they're all ones that have their own strengths and weaknesses, depending on what your application is. But like I say, this class of liquid handler, the dispenser, we found to be really powerful when it comes to doing design of experiments. And I think this video should help to illustrate that quite well. Essentially, so this is one of the dispensers I mentioned, the SPT LabTech Dragonfly. And you can see here how it's dispensing from multiple different channels simultaneously, multiple different pipettes simultaneously, without touching into the plate at all. And so it can do a complex array of different additions into the plate with rapidity and ease. So in this case, it's just using some dyes to make a pretty picture because it's a way of demonstrating how the robot works and how it can look. But this then applies equally, or maybe even more so, to highly complex design of experiments designs. And so what this means is that instead of being limited maybe to the more sort of classical number of runs that DOE has typically been used for over the years, it means that we now can start looking at some much more powerful designs of experiment, the design of experiments that can just do things that you wouldn't have been able to do previously. So there's classic experimental design DOE, which is where you have a huge efficiency. So for the number of runs that you can put in there, you can get a huge information gain, and they allow you to iteratively explore the design space, like I showed in the graph that I flashed up earlier about one of our earlier case studies of using DOE. So you're getting information about the space, you're building models, and then using those models to make a prediction as to where you could move within that space in order to iteratively walk your way up that multidimensional landscape and get a dramatically better result at the end of it. And we've seen this work time and time again. But what's really interesting, and what we've found more recently at Synthace is that, and at a good number of our clients, is that where you can use a dispenser and where the biological system that you're working with can be scaled down, so typically into, say, a 384, 96, 384, or even a 1536 well plate. So these are very small volumes per experimental run. then you can all of a sudden get way more experimental runs than we would ever have used for these more classically. We've had clients who have run 1536 run experiments, clients and collaborators, and we ourselves have run experiments with many hundreds of design points. And what that gives you is the possibility then to run experimental designs, which essentially cover the entirety of the design space very effectively in a single experiment. So it means that you don't necessarily then have to iterate in the same way, and you can get to a complete characterization of your design space a lot more rapidly than you otherwise would. We've seen the power of this at Synthace. I've seen the power of this at our clients, and it's a really exciting time to realize that this convergence of technologies of automation and then the approaches of multifactorial methods can offer us ways of cutting through the complexity of biology in a way that maybe we just wouldn't have had before. But there are some real challenges to this. So when we think about the process of running an experiment, you go through a number of different stages. You design your experiment. You think about what you want to be investigating. You then need to go through a planning stage where you're actually thinking about, okay, how am I actually going to go about running that experiment? And then you get into the lab and run it. You then need to prepare the data from it, analyze the data, and then some kind of reporting function. And if we break that down, there's a huge amount of activities that have to happen at each stage. And for a complex DOE, these can get exceptionally difficult. So we need to choose the design that we want to work with. We then need to plan the experiment. So this is all of the stocks we need, maybe the cells, the plate maps, how everything's going to map into a plate, how they're all going to be mixed together, the consumables that we're going to need to use for it, what equipment we're going to be using, is the equipment booked, is it serviced, is it working, the timings of our experiment to make sure that it's all going to fit together nicely. And then if we're going to use automation, then we're going to have to program that automation in order to carry out these highly complex experiments. And then when it comes to running the process, then we need to make up all the inputs, so make up all our stock solutions and that kind of thing. We need to perform all the liquid handling steps and then make sure that it's actually run as it should have done. And then you go into preparing the data, you gather files together, structure the data, and often you need to transform the data into a form which actually is meaningful to your biological system. And then finally, you go into statistical analysis, you build models and you draw conclusions, and you'll often draw some graphs in order to illustrate those conclusions. And those can then go into your report where you can detail everything that you did, describe what the outcome was, and publish in some form, either in the academic literature or internally as a report. Now, the bit that automation's taken care of is obviously performing all the liquid handling steps for you. But there's still a huge amount of the work which is a real pain point, especially when we're doing such complex designs of experiment. So this is like the fundamental problem, which first meant that we started building our own software within Synthace. And it's one of the main problems that our software can help to do in terms of this design and planning stage, in terms of working out all of the instructions for running things on automation, then structuring the data and helping with the analysis, model building, and recording everything, all the steps that were carried out in that experiment so that we can always go back and troubleshoot if we need to. So let me take you through that. So in Synthace, we can create or import a DOE design. So there is a capability to actually create simple designs in Synthace, but also we have that facility to import them from, frankly, much more powerful statistical tools like JMP or Design Expert or the like. Then it can generate the liquid handling instructions automatically. It can collate and structure DOE results and then get summarized DOE analysis. And I'll take you through each one of those. So in Synthace, when we're designing a new workflow, then essentially what we do is we string together these unit operations, these actions that we call elements. A lot of these elements just refer to particular actions that you might do in the lab, say mixing together some liquids. So in this case, we've got some liquids that have been defined by the user, the various different liquids that pertain to their experimental design. And we're then making mixtures of those liquids. And if you'll notice, there's a little icon on the make mixtures element, which is a little DOE icon. And what that means is we can switch to a DOE mode, and we can then list all the factors which are associated with that make mixtures element. So we can take the different input liquids and we can choose the levels that we want to investigate for those input liquids and put them into the software in a really easy way. So then what that allows the software to do is to say, okay, we can see that you want to look at mixtures of these liquids and that you want to look at them over these particular levels. And it can then generate designs for you, which will allow you to investigate those levels of liquids in a very systematic way. So in this particular instance, we have a space fill design that's been chosen. And so you can see all of the design points which are auto-generated, which can then be investigated by the experiment. But additionally, Synthace also knows about constraints that there might be in the system. So if we can't pipette below a certain volume, then it won't suggest those design points as part of the design. And obviously here we have a mixture of some factors where you can have a wide range of different points and other factors where actually there might just be two different levels for those. For example, the diluent that we're using, there are just two different diluents that we might choose. And so that then is just choosing one diluent or the other. There's nothing in between as far as this design is concerned. So once you've chosen a design, you then would want to go into the execution. So you can go straight from the design straight into then Synthace generating all the liquid handling actions which are required to carry out that design for you. We've got a number of different stock solutions which are auto-generated. The concentration of those stock solutions is worked out for you. The amount of those stock solutions that you're going to need to carry out the experiment is also worked out for you. And then critically, every single dispense or liquid handling action that's required to go from each of those stocks into the composition that's needed for each of the runs that's been defined as part of the design of experiments is then also auto-calculated. Though that sort of experimental plan is then translated into whatever format is needed for the device that you're using in the lab, be that your liquid handling device like your Tecan or your Hamilton, or the dispenser that you're using. And so then you're ready just to go ahead and execute that experiment as defined in the design stage. So you go into the lab, you run your experiment, and you can then bring together all of the results from that execution. Because we've gone through the whole design and planning stages automatically in Synthace, in silico, then that means that we have like all of the metadata of what's gone on in the experiment automatically generated. So then we can very easily append the data, which is then produced from each of those runs, to the metadata. And so it auto structures the data for you. So it's automatically bringing the end result data and bringing it and associating it to the particular design of experiments run that was generated at the beginning of the process. Now, this is a process which, from our own experience, can often take a very long time, especially for complex experiments. And it's a lot of, at its worst, copy pasting in Excel to get the data into the right format and into the right structure before you can then actually run your analysis on it to actually generate the models and see what's going on. So like this facility to just auto structure the data is a dramatic benefit. And so then, when you've got all your data in a structured format, you can then go along and do your analysis. There are all sorts of ways of analyzing DOE designs, and I'm not going to go into that now. But essentially, Synthace then just allows you to explore your data, build models, interpret what's going on in your system, and hence then move on to the next iteration of your design. So that's the overview of how Synthace goes about running complex experimental designs. So when we go back and think again about, okay, what all the issues that you might have about when you're trying to run very powerful automated experiments, then essentially what we've looked to do within Synthace is to address all of those core pain points. So then you end up with a situation where you can access this kind of much more powerful experiment without having to go through the huge amounts of pain, which frankly were pretty routine in the early days of Synthace, and I think are met by a lot of people who are trying to do DOEs. But moreover, I think what we're getting really excited about is this concept that we can explore design spaces in a much more comprehensive and effective way because of this convergence of technologies. When you have very powerful hardware. And there's now a huge selection of fantastic hardware, which, like I said before, all has its strengths and weaknesses for different purposes, depending on the system that you're working with and that you need to optimize and work with. There will be a bit of hardware which will help you to do that. You combine that with software that allows you to rapidly program that hardware to do very complex and powerful experimental designs, and then the experimental design methodology itself, so the mathematics behind it, then you end up with something which is disproportionately powerful when it comes to investigating in biological space. These technologies are not additive with each other, they're genuine synergies, because if you take any one of them away, then you get dramatic reduction in the capability that you have. You start off with one factor at a time. This is another representation of what that might look like. We've got a complex space that we're looking to explore, and we've chosen two different cross sections through that space, and we've misidentified a false maximum, a false optimum within the space. Now, we know with DOE that we can walk our way through that space. So we might start down here, but then we iterate with each successive experiment our way through that space until we get to a much better local optimum. But what we've found really exciting of late is that actually with the kind of run number that's possible and the complexity of design that's possible when you're using this kind of automation, then you can do a much more comprehensive analysis and actually identify a more global optimum within the bounds of the experiment that you've run. And critically, you can get that kind of characterization even within a single experiment. And it's just a product of the fact that there are some biological systems, particularly when we're thinking about assay development or when we're thinking about media development for complex cell line differentiation and expansion, then those are the kind of systems that we can scale down quite effectively. And so that means that we can get this kind of run number, which means that we can address biological complexity to a kind of degree that we might not have been able to experience in the past. But like, it's to a final so what? We've seen this applied in many different areas, and I wanted to give you a bit of a flavor of that. Like I mentioned, assay development is an area where this can be very powerful, particularly assay development in the early parts of drug discovery. So because the output from these experiments is intended to be as miniaturized as possible, because this is often something that would then be used for, say, a high throughput screen where people would want to run these assays maybe 1 million times, then the desired output is something that would run, say, in a 1536 well plate. So that means that you can do the assay development in a similar kind of scale. And it means that you can get thousands of data points with a very small amount of reagent and it's a very small number of plates. So what that means is that you can do these absolutely massive multidimensional experiments. What we found then is that can enable something between a 30 and 70% reduction in assay development timelines, depending on the complexity of the assay in particular. Those assays which are particularly complex, this method is disproportionately powerful compared with when you're trying to do things with one factor at a time or less sophisticated or powerful methods. So there are some very specific measures within the assay development space. So Z prime is a measure of signal to noise. It's a very stringent measure of signal to noise. And essentially, you can't go forward with an assay unless it has a decent enough Z prime. And we've seen that improved from borderline or even poor Z primes all the way through to good or even exceptional ones. And so those assays are moving forward with a much higher quality. And similarly, the assay window. So this is the sort of dynamic range of the assay, if you like, the difference between the positive and negative controls. And so how sensitive that assay can be. And we've also found that because we're characterizing the entirety of the space, it means that you can identify parts of that design space that not only work very effectively, but also might make the best use of the most expensive reagents or enable you to minimize the number of, say, cells that you're putting into an assay. And so if you've got a particularly precious cell line or primary cells, then this allows you to make best use of those very precious resources because you understand the overall assay an awful lot better than you otherwise would have done. So essentially, it means we're getting much faster development, much higher quality output, and with a reduced cost. Oh, and incidentally, one of the ways that the cost can be reduced most effectively is assays can more routinely be miniaturized all the way down to 1536 runs, which means that then if you compare that to a 384 well plate, you're essentially using a quarter of the reagents per assay run than you would otherwise have done. So that's just a very pragmatic way that you can have reduced costs, again, by having that higher quality assay that you can identify from the kind of experiment that we run. Another example is media development. We've got a particular case study that we've cited in the past, and you can find it on our website. We worked with Uncommon. So that's the company that used to be known as Higher Stake. So these are a cultured meat company, and their mission is to make it highly efficient to grow up meat in a cultured environment. So basically in a bioreactor, as opposed to using an animal to do. And their main challenge there is to drive down the cost of that media, because mammalian cell culture media is notoriously expensive. And so for that business model to ever be viable, and for that all the benefits we can have from producing meat in a much more environmentally friendly, sustainable, and humane way, the only way that's ever going to work is if we can get the media costs down dramatically. And so they use this capability to look at 22 different factors simultaneously. So this is looking at a huge number of different growth factors that are in the medium. So this was for porcine induced pluripotent stem cell expansion and maintenance. And it allowed them to map out which of those factors were having the biggest effect in their system. This was a 320 run design. It resulted in a 15-fold reduction in the media costs. So that's 15-fold. So it's a very dramatic reduction by, again, understanding this space to that level of comprehensiveness. And again, it means that because you're not looking at one factor at a time, if you're looking at 22 different factors, and each of those factors the experiment takes you a week per factor, then that's almost half a year's worth of experimentation. Whereas an automated DOE like this, although it'll take quite a lot of planning, even with the help of the tools we've been talking about in this webinar, it'll take a fair bit of thought and planning, but it's still going to be dramatically less time to conduct this substantially more powerful experiment than it would take to do a series of much less powerful experiments. So it means that the progress that you can get is expedited dramatically. And then finally, something I'd like to just call out is on the purification development side of things. We have a partnership with Tecan and Repligen, and they make this system called RoboColumns, which is a miniaturized purification system. So it's for high throughput purification development. And when we're looking at purification of biologics or viral vectors, then finding the optimal conditions for those purification is paramount, and finding conditions that are robust and that are giving us the quality and yield that we need time and time again takes a real investment of effort and time. By having this miniaturized system, it means that we can parallelize more purification runs simultaneously, and so we can get to these answers a lot more effectively. Now, programming these columns to run on a Tecan deck can be quite challenging, and the analysis of the large amount of data that comes out the other end can also be quite difficult, but we have that capability within Synthace to make that a lot more tractable. So hopefully I've given you an idea about how DOEs can be effective within biology, but then we just take it up a whole other level when we're then thinking about using automation to actually carry out those DOEs. It means that then a large number of limitations that we otherwise would have had as human beings trying to do very complex experiments then get stripped away, and it allows us to do these experiments at a kind of level of power and complexity that just otherwise wouldn't be possible. That in turn means that for all sorts of different biological applications, we can now address a much larger chunk of the complexity that's inherent in those applications. And it means that we can make much faster and more definitive progress. I've often argued that a failed experiment isn't one where you don't get the result that you wanted. A failed experiment is where you get a result where you don't know what it means. And with this kind of experiment, we're finding that actually that kind of situation is happening a lot less, and we can make much more definitive progress with our biology. With that, I'll leave it. I'll say thank you very much for your attention, and we've got some time for some questions. Okay, so we have a Tecan liquid handler and not a dispenser. Can we still run the kinds of high-powered DOE that you described? Yeah, in short, yes. There are some additional considerations that you need to think about. The advantage, obviously, of a dispenser is that you, like I said in the talk, you're not actually having to touch against the you're not having to touch against the liquid that you're pipetting into. You can do it from above the well. So what that means is you don't have those tip changes. So if you can work out, and we've done a fair bit of work about this internally in our own labs, if you can work out ways that you can get effective dispensing from above the well without having to touch again, then you're effectively turning your liquid handler into a dispenser. And I know that some liquid handlers have, a good ability to be able to do this. So although it's unlikely to be quite as effective as using something like a Dragonfly or an Echo or a Tempest, then yeah, you can still absolutely do this. And the thing is that even without being able to dispense above the well, you can still do dramatically more powerful DOEs on a liquid handler than you'd be able to do by hand. So it's a matter of degree, really. Then we've got another question here. Okay, so it's a very similar kind of question. What automation would you recommend for starting out doing automated DOEs? We don't have any automation yet. Okay, you're starting with a blank slate. It depends very much on the biological use case that you have in mind. Like I say, I can't really go into a full survey of all of the different automation solutions that are out there in the space of answering question. But yeah, essentially, you need to look into the strengths and weaknesses of the different bits of automation and choose one which is most suited to your particular applications. For example, we've seen a lot in assay development that you tend to see things like the SBT Dragonfly, Echo, and the Synthace Flex all used in conjunction because of their different strengths and weaknesses. For example, the Dragonfly is very flexible, very approachable and easy to use. And then the Synthace Flex, for example, is just super fast. Whereas the Echo is a bit slower, but can do really small volumes. So when you're miniaturizing your assay dev down to 4 microliters per run, which is what you'd have in a 1536 well plate, then that's the kind of system that you'd use in that particular instance. With that, I'll just end by talking about our DOE Masterclass series. Obviously, I've spoken in general terms about DOE in this webinar, but that wasn't the main purpose of this webinar. And so if you want to go into DOE in more detail, then that's a good place to start. We've got all sorts of different webinars, both from people internal at Synthace, and we've had some external speakers on that as well. So that was a really fun series to do. Feel free to go check it out. It's all available on demand. And then another quick plug, and that is also if you want to get started with DOE and you want to see what this could do for your own research, then yeah, check out the DOE training that we're just about to release, which is from our very own Dr. Michael Sadowski and Dr. James Arpino, both experts in the area and huge evangelists. They're known as Sid and Jar. And yeah, I'm really excited about them releasing this stuff so that you can have a nice step-by-step guide to getting started with DOE. And with that, I'd like to thank you very much for your time today and I wish you a very happy holiday season, and we'll see you in the new year. Thank you very much, everyone.